Hello, my name is Joshua Mugabi. I welcome you to Human Biology Part 9. Without wasting any of our time, let's go directly to our quiz. Why is pH important to organisms? Biological molecules, especially proteins, function based or only at a certain pH. Proteins can lose their shape and thus their function at the wrong pH. Metabolic reactions can require a certain pH in order to react. What does a buffer do? How is this important to living organisms? A buffer resists changes in pH. It does not make the pH neutral. Buffers enable organisms to control their body's pH within a narrow range. The element dash is the basis for so many organic molecules because each atom of it can form covalent bonds with 4 other atoms. Carbon. Define hydrophilic and hydrophobic and give an example of a molecule or functional group that can fit each description. Hydrophilic is water-loving. Example, hydroxyl group. Hydrophobic is water-avoiding. Example, methyl group. Name 3 types of polymers common in all living organisms. What monomers are used to build each of them? Carbohydrates are composed of sugars. Proteins are composed of amino acids. Nucleic acids are composed of nucleotides. Why are nucleic acid called the information molecules? Name 2 examples. DNA and RNA contain the information in a 4-letter code for the primary structure of all proteins in the cell. How are large macromolecules built within cells? In other words, Name and describe the type of chemical reaction that is used. Dehydration synthesis, in which two molecules are joined together and water is removed to form the covalent bond. What reaction is the opposite of dehydration synthesis? How is it opposite? Hydrolysis, in which a molecule of water is added as a covalent bond, is split apart. Glucose and fructose are examples of monosaccharides. Glucose and fructose are examples of monosaccharides and are components of sucrose. The main purpose of simple carbohydrates in the cell is energy. Name 3 common polysaccharides, their monomer, and in what group of organism each polymer is found. Starch made of glucose, made in plants. Cellulose made of glucose, made by plants. Glycogen made of glucose, made by animals. What are lipids? What property is their main characteristic? Lipids are fats, oils, and steroids. Their common trait is being insoluble in water. What are triglycerides? How are they used in organisms? Triglycerides are glycerol combined with 3 fatty acid chains. They are used for energy storage. What property of phospholipids makes them so useful in cells? Polar and non-polar ends in the same molecule makes them hydrophilic and hydrophobic, perfect for forming membranes in aqueous cells. Define denaturation. What three conditions can cause denaturation of proteins? Loss of native function, i.e. shape. Too low or too high pH. High temperature. Too high or too low salt concentration. Name another category of lipids and describe their basic functions. Steroids used for signals, that is in hormones, and membrane components. Examples, we have cholesterol. Describe at least 4 different functions of proteins. Transport, we have hemoglobin carrying oxygen. Structure, there is hair. Enzymes, example, amylase to digest starch. For defense, we have antibodies of immune system. For signals, we have human growth hormones. Nutrients, we have for essential amino acids that we cannot build ourselves. What is the basis for the diversity of proteins in cells? i.e., how can there be thousands of different proteins? Proteins are made from up to 20 different amino acids. In any sequence, if a protein is any amino acids long, there are 20n different possible combinations, i.e., different proteins of this length. A typical protein is 100 to 300 amino acids long. What is an amino acid? What purpose does their group serve? Molecules with an amino group, a carboxyl group, and an R group attached to the same central carbon atom. The R group confers chemical traits such as solubility and electrical charge. Describe 4 levels of protein structure and the factors that determine each one. One, the primary structure. We have amino acid sequence. Two, secondary structure. There we have hydrogen bonds between amino and carboxyl groups. The third one, the tertiary structure. The R groups determine overall folding. And then the fourth, the quaternary structure. This one is an association with other polypeptides. Why is the shape of a protein important? Shape determines function. If a protein loses its shape, that is being denatured, it cannot perform its function. Whenever a protein must interact with another molecule, its shape is the key to that interaction. Dear friends, thank you for attending to human biology. Please do like, share, and subscribe. Thank you. I'm Joshua Mugabi. Bye-bye for now.